All right, good morning, everyone. Welcome to the beginning of Cycle 9. I hope that the midterm exam went well for many of you. The makeup exam takes place March 20th from 6 to 8 p.m. So please make sure that you don't discuss any of the midterm exam questions on the Brightspace discussion forum or on social media or any of the chat boards until the makeup is complete. There's a new policy that there has to be significant time between the midterm and the makeup, which means that the grades will not be released until after the makeup. So I'm not sure what policies other courses are following and how it's working, but in biology, we're keeping a significant gap between the midterm and the makeup and following the policies. So I appreciate your patience, but the grades will be released after the makeup is complete. So we're going to begin Cycle 9 on Alzheimer's and then end Cycle 9 with cancer genetics. And the reason I chose these two topics is really because I would consider this to be my area of expertise. I did my master's degree in Alzheimer's or neurodegenerative diseases, did Parkinson's as well, and I did my PhD in cancer genetics, specifically breast cancer, using mouse models for my PhD and rat models for master's. I feel like I'm in a good position to really give you a lecture where I feel like I just took a dive in the ocean and I picked up all the pearls, and I'm picking up all the shiny pearls and presenting it to you. So hopefully you'll appreciate it. Lectures like these can be really given with that ooh and ah effect. And what I mean by that is you come to the lecture and you look at it and you go, ooh, ah, the before treatment, the after treatment, look what's happening. And those lectures are really great to attend and to listen to, but here's the problem. You leave that lecture and you pick up a journal article and you understand nothing. And I've been to a lot of ooh and ah lectures, and you pick up a journal articles and you understand nothing. And the reason for that is because the lecture wasn't given in the way it should have been given, which is the way I want to give it to you, so you can pick up an article, a peer-reviewed journal article, and understand the details. And you will understand at least 50% of it for sure. So buckle up, here we go. Alzheimer's disease. It's a neurodegenerative disease. Neuro is for neuron. That's the cell in your brain. It's called the neuron. Degenerative means loss or deterioration. So deterioration can mean that that neuron is still there, but it's just not functioning properly. And loss is literally disappearance of the neuron. The neuron is dead and it's gone. It's chewed up and it's all gone. So the neuron has been lost. So a neurodegenerative disorder is one where you get loss or deterioration of a neuron, and Alzheimer's disease is an example of that. Alzheimer's disease is a progressive disease, meaning once it starts, it just continues in a one-way street. You can't move it backwards. You can't treat or cure Alzheimer's. Like cancer, for example, right? You can get surgical resection of the tumor if needed, and you can get chemo, radiation, adjuvant therapy, and many people live happy lives in remission. But Alzheimer's is not like that. It's a one-way street, and it goes that way. So once you get the onset of Alzheimer's, it progresses. It's a progressive neurodegenerative disease. Now, of course, you get different ranges of severity, right? You can go from mild symptoms to severe. So mild, meaning early stages of Alzheimer's, and depending on how fast you progress, that will determine the outcome. So that has much to do with your genetic makeup and your environmental stimulus, right? So your life as you're living it and environmental factors that you're bringing into your body, as well as your predisposition for it to begin with. So both those factors come into play, and that dictates how fast it'll progress. Alzheimer's disease is the #1 cause of dementia. So dementia doesn't mean that you have Alzheimer's. Alzheimer's has very specific pathological hallmarks, which we'll see, and so if you have those in your brain, then you have Alzheimer's. But you can have dementia without having Alzheimer's. So this loss or deterioration of neurons, it starts in the hippocampus. The hippocampus is involved in memory. So storing memory, retrieving memory, long-term or short-term, which is the reason why one of the key hallmark symptoms of Alzheimer's is loss of memory. One of the first things that you see as a symptom of whether you may have Alzheimer's or not is loss of memory, because this deterioration starts in the hippocampus. It begins here and it radiates outwards in a centrifugal way and it affects the entire brain. Eventually, as the disease progresses, you don't only just get loss of memories, but you get loss of cognitive function, you get muscle impairment, and ultimately death, depending, again, on the severity of it. So you can get early onset or late onset of Alzheimer's disease. Early onset would mean somewhere between 30s to 50s. So 30, 40, 50s. That would be early onset. Late onset, past 65. So after 65, that's late onset. Early onset is usually related to genetics. So you have inherited genes that make you more susceptible or more vulnerable to getting Alzheimer's because you now have an increased chance due to your genetic makeup. So you may have inherited certain mutations in certain genes from your family tree that now leads to an increased probability because you have genetic susceptibility behind it. Late onset doesn't always have genetic susceptibility. That's just old age. Old age, you're accumulating these mutations, and that can lead to Alzheimer's. And again, late onset can also have a lot to do with genetics. So your genes and your environment are the two factors. So in case you're like, well, how come this person got it? How come this person didn't get it? What about this? You have a wide range, like any disease. So I know that I'm focusing on Alzheimer's and cancer in this lecture, in this cycle. But for those of you that are not interested in Alzheimer's or cancer, please understand the following. I'm only using these two diseases as examples because that's what I studied in. But if you understand the premise, then you can take the premise, which is your foundation, and put any disease and understand that disease. What are they? Number one, you have to understand the mechanism of the disease. What leads to that disease? What are the genes involved, and what is the molecular pathway? From that, you can deduce the targets. In other words, I want a drug that's going to treat this, so what should the drug target? Which gene, which protein, which RNA, where should it go and bind to inhibit it or activate it? And then you ultimately see how to design that drug so it can do that. And what is the outcome in the individual? So those steps are almost identical for any disease. So don't take it like, oh my God, Alzheimer's, we're wasting my time, cancer, wasting my time. I get it, it's not for everyone. But if you're interested in any disease, then this is a generic lecture also. Understand, take the same course that I took for Alzheimer's, same course for cancer, apply it to anything else. So there are really three pathological hallmarks here of Alzheimer's. You have deterioration of cholinergic neurons, formation of plaques, and tangles. So let's go one by one. What we're going to do is cover this one, how it occurs normally, and how it's deteriorated in Alzheimer's. This one normal, and how it leads to Alzheimer's, and this one normal, and how it leads to Alzheimer's. That's the three subparts to this lecture. So cholinergic neurons are neurons that are involved in memory and learning. So the way neurons work is they communicate through this area called the synapse. You have a presynaptic neuron that meets a postsynaptic neuron. They come together, and that's how they communicate. And the number one way in which they communicate is through release of neurotransmitters. When I was doing my master's, even though I was focusing on very specific diseases, I had to learn every neurotransmitter, all the neurons in the brain, how they work, because you can't just take one little part of the disease and just focus on that, because your whole brain gets affected, right? So you have to learn everything. And that was, guys, it took me a whole year to learn all of that. I was like, oh my God. And then you get tested verbally on it. So it's not as easy as it sounds, but it was fun. But anyway, neurotransmitters is the way it works. The neurotransmitters get released, and then that gets picked up by the postsynaptic neuron, and it sends an impulse. So these cholinergic neurons, they respond to acetylcholine, which is a type of neurotransmitter. And so in Alzheimer's, this is the neuron that is affected. And therefore, they cannot remember, they cannot recall things. This also affects learning. APP is a normal protein called amyloid precursor protein that sits on your membrane, and it's involved in just the normal function of a neuron. It helps the neuron function normally on a day-to-day basis. When this gets deregulated, you get the formation of plaques. How many of you guys have heard of plaques and tangles in Alzheimer's? It's the pathological hallmark. If you have plaques and tangles, you have Alzheimer's, period. So plaques and tangles. Plaques are basically these clumps that get formed, and they are formed outside the neuron, and they can interrupt with the synaptic connection. And they can also lead to the direct death of a neuron, which we'll see in a bit. Tangles are formed due to a protein called tau. Tau is involved in the cytoskeleton of the neuron. It keeps the cytoskeleton together. And once tau is deregulated, boom, you get the formation of tangles. Tangles are formed inside the neuron, and they literally just get rid of the cytoskeleton. It's like literally your skeleton being gone inside you. So that then deteriorates the neuron, it kills the neuron, literally. So this is why you get loss. You start to see loss and deterioration. So here you have a normal brain with normal neuronal connections. This is a brain of an Alzheimer's individual, which is significantly atrophied because you get complete loss of mass. If you weigh the brain, of an animal with or without Alzheimer's, you can see the difference in the mass itself because you literally have the neurons disappearing and it's just replaced with fluid. And here you can see very poor to little neural connection. Now again, these are the two extremes. There are people that are living with Alzheimer's. And if you're more interested, come to me and talk to me during office hours, but yeah, there are people that have plaques and tangles and they're living life. And there are ways that people have found to live with Alzheimer's. Like, you'd be surprised what you can do. And prevention is a really big area of medicine right now, especially for Alzheimer's, because the cure seems almost very, very difficult or almost impossible at this point. So to understand all the mechanisms that lead to Alzheimer's, you have to understand the basic structure of the neuron. Here you have a basic structure of a neuron with the cell body. It's also known as a soma. The cell body is the cell where you see the nucleus and all the organelles that you've learned through this whole course, right? You see endoplasmic reticulum, you see all the organelles that you learn. That's where all the processes occur, transcription, translation, etc. Protruding from the cell body, you have dendrites. These dendrites will ultimately communicate with the axon terminal. So you have this region called the axon, A-X-O-N, and at the end of the axon, you have the axon terminal. So the axon terminal of the presynaptic neuron, which is this one here, meets the dendrites of the postsynaptic neuron at a region called the synapse. The synapse is very important. That's where communication occurs. That's where neurotransmitters are released. When a neurotransmitter is released, then the neuron responds to that, and it sends a transduction to the next neuron, to the next, to the next, to the next, which is why what you perceive in your brain and your feet, you have that connection through neurotransmitters sending signals really fast. The impulse is very fast. So this is an electron micrograph of what a synapse actually looks like. This is your presynaptic neuron, and look at that. That whole thing is a mitochondria. So when you see cartoon pictures, I think they're really nice for first year, and you're like, oh, look how beautiful that cell looks. Everything's so proportionally right, just looks right. And then you look at the electron micrograph in fourth year, and you're like, what is that? The thing you've been studying for the last 4 years, and the stuff you got the degree in, that. Oh, right, of course. Because you've never seen one. I never saw one until my master's. I walked in my graduate school, I'm like, oh, I graduated. And you look at the scope, you're like, what is this? That's a mitochondria. Why is it bigger than the cell? You know what I mean? Like, why is it so huge? Because that's the size of mitochondria here. So just understand that things don't look the same in a textbook. So in case you want to go into neuroscience, just familiarize yourself with electron micrographs so you're not shocked when you look in it. So what we're going to do is cover cholinergic neurons, how they function normal, and how they're deregulated in Alzheimer's. Then we'll cover plaques how they are developed in Alzheimer's, then we'll cover tangles. So it's a 3-part lecture. Okay, cholinergic neurons. Let's see how they function normally. Presynaptic neuron, postsynaptic neuron at the bottom, synapse over here. The neurotransmitter that is responsible for memory and memory formation and storage and retrieval is acetylcholine. Acetylcholine is produced here in the presynaptic neuron, and then it is released and it is received by the postsynaptic neuron. So who's making it? Well, this is how it's made. It's made in 2 parts. You get choline and acetyl-CoA coming together through an enzymatic reaction, and the enzyme involved is choline acetyltransferase, or CAT. CAT puts both of these together and makes the neurotransmitter acetylcholine. Acetylcholine then leaves the cell, and it goes and it binds the receptor that is present on the membrane of the postsynaptic neuron. Neurotransmitters don't just barge through the membrane and get in there. They need to bind to a receptor. They bind to a receptor, and that sends a signal transduction for the impulse. So acetyl-CoA gets released by the mitochondria. You guys know how acetyl-CoA is produced in the mitochondria through Dr. Maxwell's lecture. The citric acid cycle, by the way, just so you know, it's not just to make NADH, FADH2. I'm sure that many of you know that. I'm not like, yes, Nikki, we know that. I mean, imagine that whole cycle just to make NADH and FADH2. It's there as a free-for-all. Everything in the cell grabs whatever they need from the citric acid cycle. Succinyl, it's the starting point to make heme. In your blood, you know my hemoglobin with the heme? You need the citric acid cycle to make it. Acetyl-CoA, to make a neurotransmitter for memory, is taken from the citric acid cycle. So understand that that cycle has evolved through millions of years for a reason, not just to make 2 molecules for respiration. So you have acetylcholine that goes and binds to the receptor, ding, 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 it turns it on, signal transduction happens. When this acetylcholine neurotransmitter has finished this job, which is to stimulate this receptor, it then gets degraded. It is degraded by an enzyme called acetylcholinesterase, or ACE for short. Why? Because you don't need it, and you can't overstimulate a neuron. If you don't have to recall anything, then it's done. That neuron is back to the way it's supposed to be. It's not releasing acetylcholine at that rate. So this gets broken down into acetate and choline. These get uptaken by the presynaptic neuron, and then when needed, it combines again with acetyl-CoA, and then makes acetylcholine again. So that's the cycle. Make it, release, send impulse, degrade, uptake, make it, release, etc. It keeps going on and on like that. So that's normal. What happens in Alzheimer's? In Alzheimer's, choline acetyltransferase doesn't work very well. CAT is not working well. It's one of the biomarkers that people use to know what stage of Alzheimer's do you have, or how bad is it, et cetera, how much CAT do you have working. So look at this. If CAT doesn't work, then individuals with Alzheimer's make very little acetylcholine, which is their reason when they need to recall something, they can't, because there isn't enough acetylcholine to trigger the cholinergic neuron to say, remember, remember where you put your keys, remember. Can't, because those neurons are not firing, because they're not able to even make acetylcholine. So if you had to pick a target, there are many targets here that you can see, but if you had to pick a target, ask yourselves, which targets would I pick for a drug design? So scientists picked acetylcholinesterase. And what they did is they said, look, if we inhibit ACE, then the little acetylcholine that is produced in these patients will stick around in the synapse longer. Meaning that when they need to recall something, the acetylcholine will be there in the synapse, as opposed to, well, CAT doesn't work, we have to make it. And they did do work with CAT as well. We only have 50 minutes for this lecture, so I can't cover everything. It'll take you a whole year to learn everything. But this is sort of the big stuff. So the target was ACE. So that's where you get your first class of drugs for Alzheimer's. they fall under acetylcholinesterase inhibitors. So there are many pharma companies that are making drugs that go and inhibit. It's a competitive world, right? You guys know, pharma companies making the same class of drugs. Buy mine, buy mine, buy mine, they're better. Multi-billion-dollar companies. And so rivastigmine is a generic name for one of these acetylcholinesterase inhibitors. Exelon is the pharma name, the pharmaceutical company that made it, named it Exelon because that's your trademark. And the pharma company, or the biotech company, is Novartis. If this was Tylenol, you would see Tylenol, acetaminophen, and Johnson & Johnson. So that's how they, and it's under the big category of analgesics. So you would actually see, this is how you see the drugs presented. But when you want to have a discussion with someone, I think the best way to do it is not to mention the pharma name, mention the generic name, or mention the class of drugs that it falls under. You just sound more intelligent. So some people may not know that pharma name, and then you might put them in an awkward position. What is that drug? Oh, it's an acetylcholinesterase inhibitor. Oh, okay, that's what it is, right? So just, it's an ACE inhibitor category, and a whole bunch of drugs fall in that. So that's how cholinergic neurons get affected in Alzheimer's, and that was a drug that's used to increase memory. Now we're going to turn to plaques. How are plaques formed, and how do they lead to Alzheimer's? So to understand that, you have to understand the normal way in which this process works. Plaques involve the deregulation of APP. So keep that in mind at the back. Now let's learn how APP works normally and what does it do. APP stands for amyloid precursor protein, and it sits on the membrane of the neuron. And what it does, all you have to know, is that it is involved in the normal function of the neuron. That's all. It's pointless to go into the mechanism of what APP does. It's involved in the normal function of the neuron. It's important. But what's important here to note is the recycling of APP. So like all proteins in your body get recycled, APP has to get recycled too. So it simply means that they have a half-life. A protein is not made at birth and it just sits there forever, for your whole entire life. It gets recycled, right? It's done. Now it's got to be broken down, and the new protein has to be transcribed and translated and put on the membrane. And that's how the cycle goes. It's literally like your recycling center. You have your blue box. You put stuff in there when it's used. You push it out the door. It gets recycled into a recycling plant, and then you get new ones, right? And that's the cycle. Exact same thing. So the enzymes involved in this recycling are alpha secretase, beta secretase, and gamma secretase. So both of these pathways are normal. Normal. You have two different ways in which you can do it. Why? That's just the way we evolved. That's your language. It's like in society, we have multiple ways of doing multiple things, right? So it's the same thing. And which is kind of good because you have a compensatory mechanism that's there too. So this is how processing works. The protein, APP, has to be broken down into pieces because they can be handled better with smaller pieces. Once the smaller pieces are there, then it can be recycled in an easier fashion. So the first step is for alpha secretase to come, and it cuts right there. When it cuts right there, it pushes that green out. That's a piece of the polypeptide chain, right? This whole thing is a protein. Then the next step is for gamma secretase to come, and it cuts precisely there. This blue is not supposed to be there. Put an X on it. This just means that this was pushed out. That's all that means. This is not two blues over here, obviously. This is X. This is pushed out. So notice how the APP processing involves pushing everything out of the neuron, which is why the reason plaques are present outside the neuron, because the process of APP processing is to push everything out. So gamma secretase and alpha secretase, they cut very specifically. You guys know how precise enzymes are, right? They go and they cut right between very two specific amino acids. Depending on what type of sequence they have, they will cut very, very precisely. It's not like a willy-nilly, let's go cut something up. It's like having a pair of scissors, and you cut exactly on that line. Not slightly before, not slightly after. So they're very specific. Beta secretase and gamma secretase are involved in this pathway, which is essentially the same thing, but it's just cutting at different points. Beta secretase cuts up here, releases that, And then gamma secretase comes in and cuts exactly where it normally does. So in this case, just go back to grade 3. You have a pair of scissors, and this is your shape. You're just cutting it. Cut here, cut here. This thing is now a whole piece, right? This is called the amyloid beta peptide. Amyloid beta peptide is probably the key word you have to know for Alzheimer's. Amyloid beta peptide is made, and depending on where gamma secretase cuts, whether it cuts slightly higher, slightly lower, you'll produce a range of peptides anywhere between 36 to 42 amino acids in length. It's the 42 amino acid peptide that leads to Alzheimer's. So focus now, we're still talking normal, guys. Just still talking normal. This is all normal. It's normal. We do produce 42 as well. We normally, without Alzheimer's, produce 42. It just gets processed. But 42 is involved in Alzheimer's. Just keep that at the back of your head. So now you have this. These are soluble. Just means that they're handled differently in the recycling center. They know what to do. They chop, 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 chew it up, distribute all the amino acids. It all gets recycled. Beautiful. We'll use it for the next one. Keep going. Same thing with this. The processing of this, however, is slightly different. This here is cleared by microglia and astrocytes in your brain. What are they? Microglia and astrocytes are the immune cells in your brain. That's what they are. So they do a bunch of things, and one of them is to get rid of this insoluble amyloid beta peptide. And it does so by the help of apolipoprotein E. Apolipoprotein E is a molecule composed of lipids and protein. A lipid sphere with a protein inside it, that whole thing is apolipoprotein E, and it goes and it helps chew this thing up and get rid of it. So this is all part of the recycling center, you can see. So microglia, astrocytes, parts of the immune cell, and they release apolipoprotein E. All together they come and they process this. Okay. So in a normal brain, you have production of this and clearance. Production, clearance, production, clearance. How is it produced? Through this pathway here. How is it cleared? Through microglia and astrocytes. That's the pathway. And you end up with a steady state level. Produced, clear, produced, clear, produced, clear. In Alzheimer's, you don't get a steady state level. What you get is increase in production, i.e. this is going faster, and decrease in clearance. Why do you get increase in production? Because individuals with Alzheimer's disease have an increase in beta secretase. They have higher expression of beta secretase, which means that you now have much more of this one going on than you do this one. And the reasons for that simply is because of another problem, right? They have a problem in the signal transduction that regulates beta secretase to say, make more beta secretase. Make more, make more, make more. Make a bunch of beta secretase. Whoa, why? Because all you're going to do is just keep making a whole bunch of this insoluble one and hope to God that your microglia and astrocytes can work really well to clear this thing up. But they can't keep up. It's like a recycling center. You have a bunch of blue boxes with a whole bunch of recycling stuff, and the recycling plant is going wonderful. You can have all the boxes you want there, but I can only do so much. We only have so many machines that can recycle. That has to wait. So while this is waiting around to be recycled, guess what happens? Each one of these, picture a whole bunch of them, they all start to clump together because they're sticky in nature. But it's the 42 amino acid one that has propensity to stick together. And when these all stack together, they form what's called an amyloid beta aggregate. It comes from the word to aggregate, to take smaller subunits or smaller sections or smaller pieces and bring them together as a whole. That's an aggregate. So that aggregate is called a plaque. So you can call it an amyloid beta aggregate or a plaque. It's the same thing. So I wanted you to know both definitions so that when you pick up an article and go, what the heck is an amyloid beta aggregate? It's a plaque. So in Alzheimer's disease, this is overstimulated beta secretase. You get a whole bunch of this pathway happening. You get a bunch of amyloid beta peptides. So I want you to know the terminology. amyloid beta peptide. That's a peptide. Whole bunch together, amyloid beta aggregate, or amyloid beta plaque, or beta amyloid plaque. Scientists like to use all different versions. They all are the same thing. Nothing is different from each other. It's just terminology that the person writing the paper decided to use. This is your plaque. Now I want you to focus on gamma secretase. This whole structure here is gamma secretase. Gamma secretase is the enzyme. Anything ending in ASE is an enzyme. It's the one that cuts here, right? Okay, so gamma secretase is made up of a protein complex. So it is a protein complex. In this example here, you see 4 different proteins: the green, the blue, the orange, and the red. All together, this whole thing is gamma secretase. I want you to focus on the orange one. The orange is called presenilin 1. That is the name of the gene which makes the protein presenilin 1. What is presenilin 1? Why do I want you to focus on that? 2 reasons. Number 1, that is the catalytic subunit of gamma secretase, meaning that is the region that does the catalysis. What is the catalysis here? Cutting. Good. Cutting. That's the catalysis, the scissors. Boom, cut. The second reason I want you to know this is because if you have a mutation in presenilin 1, you have genetic predisposition to Alzheimer's. So if you inherited a mutation through your ancestors in presenilin 1, then you have a much higher probability of getting early onset of Alzheimer's. This is why I want you to know these key molecules, guys. You pick up an article, believe me, you will understand more than half of it. So, what is the plaque doing? I hope it's obvious. I mean, imagine you have a clump, right? And these plaques, by the way, are very organized structure. It's like they stick together in, beta sheets and helical structures. So it's like a sticky pad. Each sticky pad would be an amyloid beta peptide. You put all that pad together, it's one pad. That's the plaque. So here you have the plaque. So the obvious thing that the plaque does, I hope for most of you, is you're pushing this stuff out of the cell, right? And the plaques are being made. So they're going to eventually clog the synapse. They're going to make sure that that connection between the post and the pre can no longer work well because, well, you're clogging it. It's like literally clogging a sink. You put a bunch of stuff in there, water won't flow through it. Same thing, neurotransmitters can't go through it. There's no signal happening. The plaque may be sitting on top of the receptor. The plaque may be sitting in a place where the neurotransmitter has to be released. So that, once you have this, you might as well just not have any functional neuron, because even if the neuron has its integrity and is sitting there and it can do everything, well, if it can't release neurotransmitters, and if the synaptic cleft is blocked, the neurons might as well be dead. They can't work. So this neurodegeneration here. You know what else happens in your brain when plaques are formed? You can get mild versions of this or severe versions of this. Your cell, or your astrocytes and microglia in your brain, they think that the plaque is a foreign molecule. They've never seen a plaque before. They've seen amyloid beta sheets. They're like, yeah, we know how to get rid of amyloid beta peptide, if it's a peptide one, but not when it's all clumped together. We can get rid of a sticky note, but not a whole sticky note pad. What the heck is that? So they mount an immune response. They're like, don't worry, we got this. We got you covered. We'll get rid of this plaque. And in the process, they're trying to get rid of it. They can't. So guess what they do? They mount an even bigger response. They're like, okay, this is not big enough, bigger and bigger and bigger until they try to get rid of it. And you know what that does? That whole process, that immune process, releases cytokines. Cytokines cause inflammation. So you are causing inflammation of your brain, and that in itself will damage neurons too. Like, it's just a disaster when it comes to later stages of Alzheimer's. It's an absolute disaster. This is why you see that kind of atrophy in the brain. So now you have this immune response. In addition to clogging the synapse, you have an immune response. Your brain is inflamed. You literally have inflammation in the brain that is killing the neurons. Inflammation does a lot of havoc, guys, not just in your brain. You guys know how bad inflammation can be. A lot of diseases can be actually settled down if you just control the inflammation in your body. and much of that can be done with antioxidants. So yeah, these are the two reasons why, or the two outcomes of a plaque. Now let's look at how a plaque can lead to the death of a neuron. Not just from inflammation, but a direct death of a neuron, and this has to do with something that happens indirectly. It's an indirect outcome that leads to the death of a neuron. So pay attention here. Now you have your immune cells that I mentioned, they become activated to get rid of this amyloid beta aggregate. So you have a resting microglia, which then becomes activated. So AM is activated microglia. This releases cytokines, and it activates astrocytes. Astrocytes release ApoE, apolipoprotein E, which is a lipid and protein structure. This goes, normally this just goes and gets rid of the amyloid beta peptides. When you start to get aggregates, which you mainly will because you have propensity to get Alzheimer's, so you start to get this aggregation, it does go and do a decent job in getting rid of some aggregates too. But the more you're producing aggregates, the more you're accumulating them, and these cells can only work so fast. But they're working at their maximum capacity. So just keep that in mind. They're working at their maximum capacity trying to get rid of this amyloid beta aggregate. You guys know, right, that in a cell, whenever you have a reaction, you get byproducts. So what's the byproduct of our cellular respiration? Carbon dioxide, water. It's a byproduct, right? They get just released and they get handled. The astrocyte also releases a byproduct, and its byproduct is glutamate. So normally it's like, okay, the cell goes, okay, you got some glutamate, perfect, all right, I'll use some up, and the rest can go recycled. We're good. That's normal. But what happens when an astrocyte is overstimulated and it is producing more and more and more and more glutamate constantly? What is glutamate? Anyone know what glutamate is? It's a neurotransmitter. It's another type of neurotransmitter. And so now the glutamate also has a specific neuron that it stimulates, right? It's the neuron that responds to glutamate. And its receptor is NMDA. That's its receptor. It's called NMDA receptor. It is specific for glutamate. So this neuron here, which is an NMDA receptor neuron, it now starts to accept glutamate. It doesn't have a choice. It's got a whole bunch of receptors on its membrane. And glutamate's right there. It's like ding, ding, ding. It keeps turning this neuron on, and it is overstimulating this neuron. Overstimulation of a neuron kills the neuron, period. How, when, why? That's for another course or another lecture. But I hope that's common sense. Cells have to have regulation, tight regulation. You overstimulate this thing, it's going to die. Because it goes, there's something wrong here. There's something wrong. So I won't go into the mechanisms, but there are many reasons why the neuron will die. Partly because it thinks it's protecting you. There's something wrong, die. Because something's not right. I should not be overstimulated this much. So here you go. You have overstimulation due to glutamate and the death of a neuron. So now you can see what the plaque has done. The plaque clogs the synapse. It causes inflammation in the brain due to this immune response. And it overstimulates glutamate neurons, which then lead to death of the glutamate neurons. So plaques have a lot of outcomes from a pathological standpoint. This is just for your interest. This is what an amyloid beta aggregate looks like in a test tube. That's an electron micrograph. That's what it actually looks like when these are individual amyloid beta peptides that then just aggregate together. So now you have that glutamate neuron, right? That's going to be overstimulated. So you have the presynaptic neuron, the postsynaptic neuron, and the receptor that glutamate recognizes is called NMDA, the NMDA receptor. There's a whole bunch of NMDA receptors on there, and glutamate is activating it. To understand this a little bit more detail, I want you to understand agonists and antagonists. They're mainly used in neuroscience. So you have a receptor, it binds to its endogenous agonist, meaning if something comes and binds, and it activates that receptor. it has a binding site for that receptor. Binds, turns it on. You can get an antagonist, which comes and replaces the place of the agonist, and it prevents the agonist from binding and activating that receptor. So you could see it as a reversible or irreversible competitive inhibitor of an enzyme. So that's what the difference between agonist and antagonist. So an antagonist would be something that would come and bind to the place that glutamate binds, so that glutamate cannot bind there and stimulate NMDA. It works like a competitive inhibitor. And so that's where you get the next class of drugs for Alzheimer's. They fall under this large class known as NMDA antagonists. And memantine is the most common drug that's given in this category. And this is to protect the neurons that are dying from glutamate overstimulation, because your astrocytes are working at such high levels. Okay, so this is the next class of drugs. So we covered cholinergic neurons with acetylcholine and how that gets deteriorated because of their decrease in the production of CAT. Now then we turn to plaques, and now we're going to turn to tangles. How are tangles formed? Here you have a neuron with its cell body, axon, and axon terminal. In the axon region, you have these structures called microtubules. Microtubules are essentially giving it structure. It's giving it the cytoskeleton structure. It's literally like our cytoskeleton. Not literally, but similar. So this is what the microtubule looks like. It has two subunits that are held together by proteins called tau, T-A-U. Tau can be analogous to nails or screws on a table or a shelf. If you put a shelf together with nails or screws, the nails and screws are tau. They keep those pieces together, the pieces of the wood together. If you remove the nails and screws, the whole thing's going to fall apart. That's exactly what tau does. Tau keeps these subunits together. Now, the next step is to turn into the language of the cells. Cells, as you know, are activated or inactivated, function, not function, based on their phosphorylation state. So if a protein is phosphorylated, it may work or it may not work. It just depends on the protein. But phosphorylation is their language. It's not English, it's phosphorylation is one of it. So when tau is hypophosphorylated, meaning less phosphorylation, HYPO is less, hyper, ER is more, hyper. Hyper is more, hypo is less. When tau is hypophosphorylated, meaning it's less phosphorylated, it is bound and keeps the microtubules together. That's how it functions. But there's a deregulation in Alzheimer's through another pathway that goes and adds more phosphate groups to tau. It hyperphosphorylates tau. And when tau is hyperphosphorylated, it can't function. So it releases itself from the microtubule. So it's analogous to you taking a screwdriver and unscrewing the screws, and now the table just falls apart. So you've hyperphosphorylated the screws. So hyperphosphorylation, tau dissociates. It's like, I can't work. When it does, all of these subunits fall apart, and the microtubule, guys, literally breaks down, and the neuron is dead. It's like, obviously. I hope it's a no-brainer to everybody. It's like you standing there and your skeleton disappears. I think you're dead. So this is an obvious mechanism of neuronal death, but here's what else happens. You know how you had the amyloid beta peptides that aggregate together? Well, guess what? Tau also forms aggregates. You can get tau aggregates, but seldom have I seen tau aggregates. I don't know why. It's usually tangles. They decided not to say tau aggregates, but I've seen those in journal articles too. Tau aggregates or tangles. So these two also cause very similar type of havoc inside the cell. Now imagine the cell has all of this tangles present, right? So I think before we worry about the tangles so much, I mean, look at what's happening to the microtubules itself. So plaques tangles cholinergic neurons, causing the death of the neuron, and that's leading to Alzheimer's. So now that we've covered all of this stuff, look how beautifully you will understand this slide. You can just go through this and you'll be like, I understand it perfectly. Down syndrome individuals have an extra copy of chromosome 21. APP gene is coded on chromosome 21, which is the reason why individuals with Down syndrome have a much higher probability of early onset of Alzheimer's. The numbers may have changed, but back in the day, if you had if you had Down syndrome, by the age of 40, you had Alzheimer's. Now, about, I think over 60% of individuals with Down syndrome have Alzheimer's by the age of 60 or 55 or 60. So it's pretty common. Why? Because the APP gene that is being processed, right, by beta secretase and gamma secretase to make that amyloid beta peptide, you just have more of that happening now because you have an extra copy. So more probability that you're going to accumulate and accumulate and accumulate these sticky versions, and through over time, you can see the plaques in individuals with Down syndrome. Mutations in presenilin 1 and 2. Presenilin 1 is coded on chromosome 14, presenilin 2 on chromosome 1. Do not memorize the chromosome numbers, that's just memorization. I'm never going to ask you, you can always Google that stuff, but know the rest of it. This is found in familial Alzheimer's disease or early onset. So what this mutation is the following. It's mutation in presenilin 1 in such a way that it prefers to cut and produce the 42 amino acid amyloid beta peptide. So it's a mutant gamma secretase that produces a larger version all the time. It preferentially produces a larger version, which means that you have more propensity to make the aggregate. Inheritance of ApoE4 variants. So ApoE, that apolipoprotein E that we talked about, that is released by the astrocytes and goes and it handles that beta amyloid aggregate, you have 3 versions in the population. Remember we had different versions and variants of mRNA? 3 variants. Variant 2, 3, and 4. ApoE gene itself is coded on chromosome 19, just for your interest. ApoE2 version is rare. Rare meaning like 5% of the population have E2. And when I say population in the world, not like a specific ethnic group, population in the world, 5%. ApoE3 is the most common variant in the population, and it is efficient at removing amyloid beta aggregates. So most of us in this room have ApoE3, literally working in your body right now to get rid of any aggregate that is being formed in your brain right now, ApoE3, and it is efficient at it. ApoE4, though, guys, is a variant that you can inherit from your parents that is not good at removing the amyloid beta aggregates. And so this mutation, if you have, it is inherited in autosomal dominant form. If you have 1 copy, if you're heterozygous, you have a 5-time increased chance of getting early onset of Alzheimer's. If you have 2 copies, 1 copy from each parent, so if you're homozygous for E4, you have a 12 times increase in getting early onset of Alzheimer's. Those are high numbers. There was an A-list celebrity. He was working on a film. This is published all over the internet. He was working on a film, and I don't know why he had to get genetic testing. I didn't read the details. The bottom line is he found out during filming that he's homozygous for E4. That was some scary stuff to find out, right? So here you have an atrophied brain, normal brain. You can see the plaques in blue, the tangles in red, plaques outside the cell, tangles inside, complete atrophy. These neurons are literally gone, and what do you see? You see just fluid there, okay? You can see all the drugs coming down the pipeline, phase 1, phase 2, phase 3 clinical trials, disease-modifying biologic drugs, disease-modifying small molecules, and symptoms. So what did I cover in this class? Look at the red. The red got to all the way to phase 3. The red is targeting the beta amyloid plaque. The dark blue is targeting tau. The yellow is the inflammatory response. So this tells you how much drugs are coming down the pipeline. And look at how many of them are just under symptom control. Because Alzheimer's is progressive, there's no going back. At least give these patients a good quality of life by relieving their symptoms. So I hope that some of you in this class will discover the cure for Alzheimer's someday. And if you do, I better be the first to get your autograph. Take care, guys.